So basically, my name is, first of all, I would like to thank the organizer for this opportunity. Right, okay, I would like to start my presentation with the title, Gellan Gums Incorporating Titanium Dioxide Nanostructures Biofilms for Wound Dressing Applications. I am Mohd Hasmizam Razali from Universiti Malaysia Terengganu, Malaysia. Right, so before we go detail, I'd like to brief a little bit about the wounds. Okay, right, a wound is the type of injury in which the skin is ripped and torn apart. Right, so they have a few examples here. We have open wound and closed wound. Okay, so and that to heal this wound, we have to come out with a wound dressing. What is wound dressing? Okay, so dressing that used to cover the wound to provide the optimum environment for the wound healing and prevent bacteria infections. So we call as a wound dressing. Okay, in our research, basically we focus more on wound dressing thin films. Okay, in this figure, you can see some example, right, of wound dressing thin films, very clear and transparent wound healing. That means we can observe the healing process over the times. In general, an ideal wound dressing, it can absorb the exudates, maintain the moisture, facilitate the wound care to cure from the inflammations, granulations, reduce the tissue damage during remodeling stage. Okay, so the dressing material source usually come from nature. We have alginates, chitosan, gellan gum, and others, or sometimes we have synthetic wound dressing. Sometimes we call it bandage. Cotton wool lint goes with a variety of dissolving degree function to manage the wound. Okay, in our research, we focus on gellan gums, one type of biopolymer. Okay, was discovered in 1977. Okay, gellan gum is one of polysaccharide that produced from fermentations of Plasmodium elodie. All right, this is the chemical structure of the gellan gums. Okay, we can see the combination of ring structure, right? So gellan gum has been used as scaffold material for tissue engineering and wound dressing application all over the years. Somehow, the limitation or the problem with the GG thin films, the pure GG thin film as wound dressing is still limited. That means the performance of GG thin film toward cell proliferations and wound healing is still low. Okay, therefore, in our research, the inclusions of nanomaterial into biopolymers GG has been reported to improve cell biocompatibility, antibacterial and antiviral properties. That's why in our research, okay, in our study, we try to add in nanomaterial in gellan gums. Okay, so what is the nanomaterials? So nanomaterial, that's mean the material that have at least one dimensions in nanoscale size. Okay, and then the nanomaterial itself can comes in various shapes. So they have a 2D, that's 0D, zero-dimensional nanostructures material. For example, we have nanoparticle, quantum dot. All right, we have a 1D nanostructured material. For example, we have nanorods, nanowires, or nanotubes. And then we have a 2D nanostructures material, come out within thin films, plate, sheet. And then last but not least, we have a 3D nanostructured material in bulk form. Okay, so in our research, we'll focus on 1D nanostructured material, which is nanotube. Okay, this is we present in figure form. You can see 0D in spherical shapes, 1D in elongated and hollow shape, 2D we have in sheet shape, and then 3D we have come in a bulk shape. Okay. And then our research will focus on titanium dioxide materials, since this is a most studied metal oxide due to their unique physical, chemical, and optical properties with high photosensitivity. Basically, titanium dioxide has been used in many industries such as food industries, cosmetic, and bakery. So that's mean we can try to use this titanium dioxide material as biomedical application incorporated in GG biofilms. So how we prepare the titanium dioxide nanomaterial? 1D for the first step. Okay, this is titanium dioxide titanium dioxide particle commercial. We put in a autoclave. We put in the furnace for 24 hours at 150 degrees C. Wash, and then we collect that sample. After that, we calcine the sample to produce titanium dioxide nanotubes. Okay. And then next, we incorporated the titanium dioxide nanotube in Gellan. 1 gram of GG, Gellan gums, to be dissolved in 100 ml distilled water, and then we put in 0.01 gram prepared titanium dioxide nanotubes, okay, glycerin, CaCl2, in GG solutions. All right, for the information, glycerin and CaCl2 as a plastic oxidizer to produce in this film. All right, so, and then we stir, and then the solution will be stirred in petri dish and put in an oven for 50 minutes for 24 hours to produce this very nice film. Okay, and then that film will be characterized using FCR, SRD, and ACM for their physical and chemical characterizations. Okay, and then for biomedical application, that film must be tested for antibacterial study first. So in this research, we use an equivalent of aureus and then E. coli as a bacterial substrate in order to study how good that our film to these two bacteria. And then for in vivo study for wound healing, we use the rats, okay, as the sample. And then the rat will be removed, the food for the rat will be removed, and then we create 8 millimeter wounds using by puncture. And then that wound healing will be paced with our synthesized biofilms. Okay. So and then the healing process or wound contraction was observed every day until the complete healing. And then we move to result and discussion. First, we characterize to confirm the formation of titanium dioxide nanotubes. Okay, this is the precursor. We use titanium dioxide powder. We have a measure. We observe that 160 nanometer of the commercial. And then this is EDX analysis to confirm titanium and oxygen elements presence. After that, we characterize the formation of titanium dioxide nanotube. Okay, we can see very nice hollow elongated nanostructures. That means titanium dioxide nanotube was successfully produced using this method. Okay, we use a TiO2 powder commercial precursor, and then we come out with this very nice titanium dioxide nanotubes. And then we come out the mechanism how titanium dioxide nanotube was produced. Of course, in the first place, we have titanium dioxide react with sodium hydroxide to produce the titaniate, sodium titaniate with layer forms. And then during the washing process, we have ion exchange reactions. We produce hydrogen titaniate nanotube at the first place, and then after heating process, hydrogen titanium nanotube to titanium dioxide nanotubes. Therefore, we can see very nice nano elongated nanotubes under them, and then we're using to prove the formation of the titanium dioxide nanotube. We use TEM, transmission electron microscope, and again we can see very nice hollow elongated nanostructure here to confirm the formation of titanium dioxide nanotube. Okay, So this is the physical properties of film. This is original GG film without the addition of titanium dioxide nanotubes. And here we have the films GG plus titanium dioxide nanotubes film. The different here, the physical form, and then we test using UV spectra. We can have the improvement in transmission rate. Okay, as usual, when we include the titanium dioxide nanoparticle in that films, the identical transmission rate was reduced to 95% due to the presence of titanium dioxide nanotubes. Okay, and then basically the diameter, the thickness of the both films are almost similar, within 9 cm in diameter and 30 micrometer in their thickness. Okay, and then we come up with Ftl Spectra in order to study the chemical properties of both films, and we can observe that the Ftl Spectra for both film are almost similar. Okay, we can see the pattern of Ftl Spectra. This is the main functional group for both sample. We can see the presence of titanium dioxide nanotubes. And this is again is a SRD pattern of titanium dioxide nanotubes. This one, titanium dioxide nanotube belong to anatase TiO2. And then we have Gellan's gum patterns, this GG, the pure GG film. And this we incorporated titanium dioxide into GG film. All right. So basically, we can observe the titanium dioxide pattern in GG plus titanium dioxide nanotube, since we only use very small portions of titanium dioxide nanotube in this film. Okay. Right. So next we see the surface morphology of both film, right, using scanning electron microscope SEM, and then we can observe here very nice surface morphology for GG films, for pure GG film. Okay, very nice surface, very smooth surface. And then for GG plus titanium dioxide nanotube film, we can see the presence of elongated structure. Okay, some kind of very the rod shapes of film. That's mean where that film is generated on that film. So that's why we can observe the very large nanorod shape of nanotube in this film. Right, again, and they just confirm the presence of titanium dioxide nanotube onto the film. Okay, so and then this one is antibacterial property test. We use two bacteria, Staphylococcus aureus and Ehrlichia E. coli. So basically, based on the results, we can say that this GG incorporating titanium dioxide nanotubes inhibits the growth of both bacteria. All right. If we observe the control here, we use as penicillin to compare, okay, as a standard sample to compare with our sample in terms of their performance to kill or to inhibit the growth of bacteria. Okay, here we can see some inhibition zone for the control sample, and then also again we can see the inhibition zone for GG plus titanium dioxide nanotubes biofilms. Okay, around 11 millimeter. Unfortunately, for pure GG film, we cannot observe any inhibition zone. Right, same result goes to E. coli bacteria, and interestingly, the exhibition zone of our sample GG incorporating titanium dioxide nanotube is larger, around 14.3 millimeters compared to the pure sample. It is control sample penicillin is only 40.0 millimeter. That means we can propose that we can prove that our sample GG incorporating titanium dioxide nanotube exhibit or shows better antibacterial property compared to control sample. That means the control sample is a very famous commercial sample, which is a penicillin. Okay, this is the result of in vitro study analysis for cell proliferations. We use a control sample. Again, we have a control sample. We have a GG sample, the pure sample, and then we have a GG incorporating titanium dioxide nanotube sample. And then after 3 days, we can see that the cell proliferation of that most of the area of our sample GG plus titanium dioxide nanotube biofilms consists of the cell. The green color is the cell, represent the number of cells. That means the number of cells very high in our sample compared to GG as well as in control sample. Next, we study the performance, okay, of our sample GG plus titanium dioxide nanotube for wound healing. Okay, this is we have the original rat. Okay, when we create the healing, we can create the wound in the body of rat. Okay, we create 2 wound area in this rat. Okay, the control sample, the control experiment, self-healing process without wound dressing. And then the second sample, our sample GG plus titanium dioxide nanotube thin film wound dressing was applied on strangled rat's wound for faster healing process. Okay, so this is when we use our biofilms, titanium dioxide nanotube incorporated in GG based on the wound. This is what we observe in these results on rats. Okay, for the control sample, that means without present any biofilm. Okay, the rats only was let to heal by itself. Okay, day 3, day 7, day 70, we can see that the wound, the area of wound becomes smaller. And interestingly, when we use our biofilm GG incorporating titanium dioxide nanotubes biofilm, what we can observe that the healing process or the healing rate is higher. All right, so, and then the specialty of the biofilms, that biofilm will be degrade by itself in the healing, and then the wound will be recovered almost 100% after 40 days. And then at the same time, we cannot see any scar left on the rat. All right, so that's mean we can conclude that our biofilms was not only capable to heal that wound, but at the same time can remove the scar of the wound. That the specialty of Gellan gum GG incorporating titanium dioxide nanotube biofilm. All right. Okay. So lastly, as I would like to claim that we can prove that a novel, that means our first novel GG incorporated or incorporating titanium dioxide nanotubes thin film was successfully prepared. Okay, and we have prepared then characterize that biofilms using FTIR, UVVs, SRD, and SEM analysis. And then we was carry out the antibacterial studies, and then the result proved that GG incorporating titanium dioxide nanotubes thin films have high antibacterial activities against E. coli and aureus bacteria. So facts, it almost similar or comparable with the standard or the control sample of penicillin, which is a very commercial, very glamour commercial antibacterial testing material, which is penicillin. Okay, and the last but not least, in vivo evaluations in vivo evaluations on rats confirmed that the thin film wound dressing could enhance the wound healing process. All right, as we discussed earlier, within the 3 days that the rats was healed. Okay, the wound on the rats was healed 100% after 40 days, and then the scar was disappear. We cannot see any scar of the wound on the rats after 40 days. All right. So this is some references was used in the study in the slide. Okay, this is my contact number, contact email. And last but not least, I'd like to thank for your attentions, and then I'd like to thank the organizer for this opportunity. Now, with that notes, I would like to end my presentations.